Hi everyone, welcome to Unit 3. We're going to be talking about the cellular level of organization today. We're going to do the first half of this lecture this week, and next week we'll finish it off. So cells. Cells are the smallest living units in our bodies. They make up all of our tissues and our organs, and therefore our organ systems, and they're what makes us us. Prior to microscopy being invented, they could not be examined because they are so small. There are some cells that are large enough to be seen by the naked eye, such as the ovum, the egg that females produce. Microscopes were invented about 400 years ago, so that's about the time that we started looking at cells. Our body contains trillions of cells. It's pretty crazy to think about. The study of cells is called cytology, and then when we incorporate biology, chemistry, and physics all together and integrate them, that's what we call cell biology. There are two different types of cells. We have sex cells and somatic cells. Sex cells are also known as germ cells, and these are our sperm or ova. Somatic cells are any other cells in the body. Somatic means body, so body cells, anything else other than sex cells. There's also cell theory. So cell theory states that cells are the building blocks of all organisms. They come from pre-existing cells. They're the smallest units of living things, and they maintain homeostasis at the cellular level. So it's good to know those components of the cell theory. And before we start looking at some of the components of the cell itself, I think it'd be good to look at what the cell looks like. So this is what a human cell looks like. This yellow portion is the plasma membrane, which we're going to talk about in detail today. Inside the cell is the cytoplasm, all of the liquid material that kind of holds the organelles in place. The organelles are tiny organs that make the cell work. Els means tiny, tiny organs. So we'll talk about all of those in depth as well as we go forward. So let's start off with the plasma membrane, that outer layer of the cell. It's the outer boundary. It's not rigid or uniform. It has a flexibility to it. Some layers are thicker than others. It is selectively permeable, meaning that it allows some things into the cell and it keeps others out. Some of the functions of the plasma membrane include physical isolation by separating the inside of cell, the cytoplasm, from the extracellular fluid outside of the cell. It also has regulation of exchange with the environment because it acts as a gatekeeper, selectively allowing some things in and some things are kept out. It has sensitivity to the environment, as the plasma membrane is the first part of the cell that's affected by any environmental changes that are happening. It also contains receptors for cell signaling, so cells can communicate with each other via their plasma membranes. And it also has structural support. It provides this stability to the tissue. Within the plasma membrane, there are lipids, membrane lipids. They compose about 42% of the plasma membrane weight, and they form the phospholipid bilayer. So the phospholipid bilayer is what gives the plasma membrane its unique properties, because it has hydrophobic tails pointing toward the inside and hydrophilic heads pointing toward the outside. So it allows certain polar molecules to travel through. Certain molecules will need channels or carrier proteins to get through the plasma membrane because of these hydrophobic and hydrophilic components. The lipids involved in the plasma membrane include cholesterol, steroids, some other lipids. There's some proteins and glycolipids within the plasma membrane as well. So let's talk about the membrane proteins. They're actually more abundant than the membrane lipids. They compose about 55% of the plasma membrane weight. So there are many different types of membrane proteins. There are integral proteins, which cannot be separated from the membrane without damage to the plasma membrane itself. Most span the width of the entire membrane. Peripheral proteins are bound to the inner or outer surface of the membrane. They can be easily separated from the membrane without destroying it or damaging it. Both integral and peripheral proteins can sometimes be confined to specific places on the plasma membrane known as racks. Anchoring proteins attach to the membrane, and they attach the membrane to other structures to help stabilize its position, just like an anchor would. Recognition proteins are also known as identifiers, and they determine self from non-self, so that when your white blood cells are traveling around destroying foreign invaders like viruses and bacteria or cancer cells, your body knows to destroy them and not your normal tissue. Enzymes can be integral or peripheral. They catalyze reactions in the cytosol or extracellular fluid depending on where they're sitting. Receptor proteins are sensitive to specific molecules or ions known as ligands or ligands. You can pronounce it either way. And when they react with ligands, they can cause changes to the cell. Carrier proteins bind to solutes and carry them across the membrane. And channels or passageways through the plasma membrane typically found within integral proteins. Channels can also be gated, where they open and close to regulate the passage of specific substances. Membrane carbohydrates make up about 3% of the plasma membrane weight. The glycocalyx is made up of proteoglycans, glycoproteins, and glycolipids. The glycocalyx extends beyond the outer surface of the plasma membrane, and it forms this thick, sticky layer to help protect the plasma membrane. It helps to anchor the cells in place by being sticky and viscous. Glycoproteins and glycolipids can also function as receptors, which allow for this specificity in binding. And they also function in recognition and in determining self from non-self. The cytoplasm is within the plasma membrane. It's all of the liquid and jelly-like material that's holding everything in place. The cytoplasm contains cytosol, organelles, and inclusions. The cytosol is generically just the intracellular fluid. There are organelles within the cytoplasm, and like I said, that means tiny organs. So these are what are going to make the cell run and perform its functions. Organelles are going to differ depending on the type of cell that we're looking at. Some cells will have different levels of organelles than others, depending on their function. So some might have more mitochondria if they need to make more energy, like skeletal muscle cells. Each organelle has its own specific function, and we'll go through each of the organelles as well. Organelles can be separated into non-membranous and membranous organelles. Non-membranous have direct contact with the cytosol, membranous do not. And inclusions are masses of insoluble materials. Some examples would be glycogen granules, pigments, and lipid droplets. So cytosol, remember, is our intracellular fluid and how it differs from the extracellular fluid surrounding the outside of the cell. So potassium concentrations are higher in cytosol than in extracellular fluid, and sodium concentrations are lower in the cytosol versus extracellular fluid. Cytosol also has more suspended proteins and less macronutrients that are just floating around. So here's a list of some of the non-membranous organelles that we are going to go through. So the cytoskeleton provides a framework for strength and flexibility. It's exactly how it sounds, the skeleton of the cell. So we'll talk about each of the components of the cytoskeleton, the microfilaments, intermediate filaments, and microtubules. So microfilaments are the smallest of the cytoskeleton. They anchor the cytoskeleton to integral proteins. and they determine the consistency of the cytosol. An example is actin. Actin is a protein found in skeletal muscle. It interacts with another skeletal muscle protein called myosin, which creates muscle contractions. Intermediate filaments are called that because they're kind of in the middle as far as size is concerned for these cytoskeleton components. They strengthen the cell and maintain its shape, they stabilize position of the organelles, and they stabilize position of the cell in respect of the surrounding cells. An example is keratin that we find in our skin and hair. It allows for waterproofing and stability. Microtubules are the largest components of the cytoskeleton. They're built from a protein called tubulin. They form the main portions of the cytoskeleton because they are so large. They can change the shape of the cell and assist in cell movement. They serve as a monorail system to move vesicles and organelles through the cell, and we'll talk about that more later when we talk about vesicles. They distribute duplicated chromosomes during mitosis, and they form centrioles and cilia, which we will also talk about in a second. Microvilli are these finger-shaped projections that arise from certain tissues, especially those that are specialized for absorption. So think digestive system. We'll find a lot of microvilli. They increase the surface area, or the SA, of a cell that is exposed to the extracellular environment to allow for these areas of absorbance. These are non-motile. You will not see these moving, but they are just projections from the tissue. Centrosomes and centrioles. So centrosomes are the mTOC of the animal cells, or the microtubule organizing center. They surround centrioles in cell division, so they are the heart of the cytoskeleton. Centrioles are going to lie perpendicular to each other and generally occur in pairs. The 9+0 array is talking about how the microtubules are arranged to form these centrioles. They form the spindle apparatus and basal bodies that are needed for mitosis. They're not present in red blood cells, skeletal muscle cells, cardiac muscle cells, or neurons, which is why these cells do not divide regularly. Cilia are hair-like extensions that arise from cells. There can be non-motile cilia, also known as a primary cilium. The primary cilium detects any environmental differences. And then there's motile cilia, which is found in the respiratory and reproductive tracts. Motile cilia kind of help move the cell, or to in things like the respiratory tract, it helps to bring up any toxins that are found in your respiratory system. So any dirt or dust that you inhale, it kind of helps to bring that up so that you can cough it out. So if you are a smoker, you might notice that you are unable to get a productive cough, and that is because the cilia that are in your respiratory tract have been either paralyzed or destroyed. Motile cilia have this 9+2 array, which again is just how the microtubules are arranged. They're anchored to a basal body. During the power stroke of movement for the motile cilia, the cilia are stiff and they are flexible in the return stroke. Flagella are whip-like extensions that come off of the cell. In human cells, they will only have one flagellum. They also have a 9+2 array for microtubule arrangement. There's actually only one human cell that has a flagellum, and that is the sperm. Ribosomes are responsible for protein synthesis. They are going to vary depending on the demand for proteins. There is a small ribosomal subunit and a large ribosomal subunit that have to join together with mRNA in order to create these proteins. Ribosomes have their own RNA, ribosomal RNA, rRNA. That's where the mRNA will attach to start creating these proteins. Ribosomes can either be free and free-floating throughout the cell or affixed to the rough endoplasmic reticulum, which we'll talk about in just a few moments. Proteasomes contain this protein-digesting enzyme known as a protease. So when proteins are damaged or denatured, they'll be tagged with a molecular tag known as ubiquitin for recycling. Proteasomes will pick up this tag and destroy said proteins with proteases. So they play a key role in immune response if proteins are damaged or denatured due to viruses or bacteria. So this is where we'll pick up next week. We'll start membranous organelles and start talking about how cells can communicate.